Operon can be compared to the smart kitchen of E. coli. Imagine E. coli as a clever chef running a tiny restaurant inside the cell. Its favorite food is glucose, quick, easy, and energy rich. But when glucose runs out, the bacterium must switch to lactose for survival. The problem is the recipe book for digesting lactose is locked away and opens only when lactose itself arrives. That beautifully regulated genetic system and is called a operon. And the core concept that is there is the operon is an inducible operon, meaning it normally remains off and turns on only when lactose is present. It works most efficiently when glucose is absent. And this system demonstrates one of nature's smartest strategies. Do not waste energy producing enzymes unless they are truly needed. Thus, the operon is a classic model of gene regulation in prokaryotes. And the structural component that is there is the gene lacZ, which produces beta galactosidase that breaks down lactose into glucose and galactose, and the gene LAC-Y, which produces permease that transports lactose into the cell. And another gene is LAC-A, which produces transacetylase, which is involved in detoxification. And promoter is there, which is the binding site for RNA polymerase, and operator is also there, and which is the molecular on-off switch. And LAC-I gene produces the repressor protein. So the structural gene LAC-Z, LAC-Y, and LAC-A are the functional genes. If when they function together as a coordinated lactose producing unit, then the utilization of lactose takes place. So the mechanism of regulation is very much important for us to understand. So when lactose is absent, what happens? In the absence of lactose, the bacterium avoids unnecessary enzyme production. The repressor protein produced by the LAC-I gene binds to the operator region and blocks RNA polymerase from moving forward. As a result, transcription does not occur, enzymes are not produced, and the parent remains off, and the factory stays closed. And now, next is what happens when lactose is present. When lactose enters the cell, a small portion converts into allolactose, which acts as an inducer. Allolactose binds to the repressor and changes its shape, causing it to detach from the operator. Now, RNA polymerase can proceed with transcription and the lactose digesting enzymes are synthesized and the parent turns on. So we should also understand, and that is very necessary for us to know, is the effect of glucose, which is known as catabolite repression. Even when lactose is present, bacteria still prefer glucose because it is more efficient energy source, as I already told. When glucose levels are high, cyclic adenosine monophosphate level, that is a cyclic AMP level, remains low. Catabolite activator protein, that is CAP protein, stays inactive and the transcription remains weak. When glucose becomes scarce, CAMP concentration rises, CAMP binds CAP protein, and the CAP-CAM complex binds near the promoter. So the RNA polymerase binds efficiently. This leads to strong transcription of operon. Thus, maximum enzyme production only occurs when lactose is present and glucose is absent. A operon can be compared to a highly organized factory, because the factory component biological equivalent can be compared to a security guard to repressor protein and a special entry pass to allolactose and the main gate as operator and the factory manager as CAP-CAM complex and the workers as LAC-Z, LAC-Y and LAC-A genes. The security guard keeps the gate locked until the correct pass appears until the manager authorizes full production only when the preferred fuel source is unavailable. Now we should know the significance of operon until closing the chapter, and that is the operon was the first clear explanation of how genes can be regulated according to the environmental condition, and its discovery helped scientists understand gene regulation, control protein synthesis, cellular adaptation, energy conservation, molecular response to environmental signals. It also became the foundation for many applications in biotechnology and recombinant protein production. And the historical importance that is there is Lac-Operon model was proposed by Francis Jacob and Jacques Monod. whose work revolutionized modern molecular biology. So, black Operon is more than a bacterial survival mechanism. It is a lesson in intelligent resource management and respond only when needed, conserve energy, adapt wisely to changing conditions. And even a microscopic bacteria teaches us that efficiency is one of the greatest principles of life. Thank you very much.